Thank you very much. Thank you for the kind introduction. So I put it as a question mark, but obviously the answer is yes, otherwise I would not give the talk today. So if the coffee has not kicked in yet or you are wandering around, this is the only thing you need to remember. My point is yes, Captopril can attenuate microglia reactivity and improve social behavior in what might pertains to autism spectrum disorder. So this is very much familiar to all of you, but my point here is that the rising incidence of autism and the considerable phenotypic heterogeneity supports a multifactorial etiology involving both genetic and environmental factors, and one of these environmental factors involves the neural environment. And in the past decade or so, I've been focusing on maternal brain reactive antibodies, which is an overlooked and very much a promising field of research, which represent possibly a modifiable risk factor that may account to maybe up to 20% of the cases of autism. So just a little bit background is that maternal antibodies, and specifically subclass IgG, begin to cross the placenta around the second trimester of the pregnancy. IgG, maternal antibodies can reach all fetal tissues, even the brain, and although the blood-brain barrier blocks the IgG entry into the adult CNS, in the fetus the IgG can cross the placenta and can gain access to the fetal brain before the blood-brain barrier has been fully developed. So in the largest studies that has been done so far, we analyzed over 3,000 plasma, and what we have found is that 10.5% of the plasma of mother of children with autism display very strong reactivity to mouse brain antigens compared to 2.6% plasma of women of childbearing age. And below you can see such an example here. Does it work? Yeah, it works. Okay. So below you can see an example of such plasma showing very strong reactivity to mouse hippocampus and control plasma showing no such binding here. And several other groups, so we are not the only one who has done those kind of work, and several other groups, including Judy van der Waters and Paul Ashwood and others, have independently identified strong association between maternal antibody reactivity towards fetal brain proteins and autism diagnosis in the child. So in order to ask what these antibodies are binding to and whether they can actually lead to pathogenicity, we have pioneered the use of monoclonal IgG for mothers with anti-brain antibodies and a child with autism. So I'm not going to focus on the left side here, how we actually clone those antibodies, but what I want to show you is that one of those monoclonal antibodies were binding a hippocampal sections where Caspr2 is expressed, but not with Caspr2 is deleted. So this antibody binds a protein named Caspr2, which might ring a bell for you because the gene that encodes Caspr2, CNTNAP2, is actually autism risk gene. And maybe most importantly is that the anti-Caspr2 antibodies are frequent in women with anti-brain antibodies and a child with autism. So just briefly, what is actually Caspr2? Caspr2 is expressed early in development, and you can see here the mRNA levels expression. Its expression is similar between males and females, and it expressed mainly in the brain but not in the placenta. And in fact, it isn't hardly expressed anywhere else in the periphery. And during development, it expressed both by excitatory and inhibitory progenitor cells. In terms of the function, well, its function was mainly characterized on myelinated axon, but that happens after postnatally, which doesn't explain why it's expressed during development. So we don't know a lot about it, but we do know that from work from Dan Gashwin and others, that the deletion of the genes that encode Caspr2, CNTNAP2, which I just told you it's a risk gene for autism, leads to autism-like phenotype in mice. There is some evidence that this protein has a role in neuronal migration, and more so, Caspr2 may stabilize dendrites and spines on excitatory cells, and deletion of this of Caspr2 actually lead to reduction in dendrites and spines. So I said that the monoclonal antibody binding Caspr2, what about its actually pathogenicity? And then in our first model, what we did, we injected the monoclonal human anti-Caspr2 antibody to pregnant mice at the time when the antibody can cross the placenta and reach the developing brain. And what we have shown is male but not female mice showed autism-like phenotype in terms of brain morphology, as well as in terms of behavior, including increase in repetitive behavior, inflexible learning, and deficits in social interaction. So what I'm going to focus about today is more of our recent work, and in this model now we have been utilizing a more physiological model, the resembling better the human condition, in which now we have dams are harboring polyclonal anti-human anti-Caspr2 antibody throughout gestation. So in this model, we immunize the mice with Caspr2 prior to pregnancy. We immunize with the extracellular portion where we know that the human monoclonal antibodies are binding. And then 2 weeks after the last boost, we put them to pregnancy. We put them together with unmanipulated male mice. Control mice receiving exactly the same adjuvant treatment, immunization, but without the protein. And again, what we found is that male but not female mice show the autism-like phenotype very similar to what we have shown before with the human monoclonal antibodies. And what these bars is actually represent here is that the titers of the anti-Caspr2, the titers for Caspr2 in the fetus is similar between males and female, and therefore that cannot explain the male bias. And if we now dissect the hippocampus for the male mice, we run single nucleus RNA-seq, we find that many of the differential expressed genes that between anti-Caspr2 mice and control mice are actually overlapping with a risk gene for autism, specifically in the CA1, in the GABAergic cells. And this further validates our model and its relevancy to autism. So 2 observations have led us to study the role of the microglia. In the first one, we published that male mice exposed in utero to the anti-Caspr2 IgG show decreased dendritic arborization and reduced dendritic spine density on pyramidal cells in the CA1 region of the hippocampus compared to mice that exposed in utero to the control IgG. And now we're going to call them the anti-Caspr2 mice and the control. And this could be mediated by excessive pruning by microglia. So what you can see here, this is a tracing of CA1 pyramidal cells. And you can appreciate that it has less branches, which it means less dendrites compared to its control mice. And you can see that if we now quantify it using a shell analysis, the length of the dendrites as a function for the distance of the soma, you can see a reduction in the complexity. So it has less length of the dendrites. And more so, we found less number of spines on these dendrites. And this is farther, actually, this is in parallel to some of our single nuc RNA-seq, when we actually find reduction in excitatory postsynaptic transcript, including HOMR1. And again, true for males, but not for female mice. So the second observation that have led us to look at microglia, we have done a microPET scan of anti-Caspr2 and control mice, and we found increase in the PBR28 signaling, which is a second generation of TSPO ligand. And TSPO is thought to be a proxy for neuroinflammation. And what you can see here is a box and whisker, and then the scatter plots, and each point here represent one mouse form a unique leader. And TSPO is thought to be it's a mitochondrial protein that is expressed on microglia and astrocytes, and its increased expression could reflect increase in neuroinflammation and or mitochondrial dysfunction. And obviously there's not a lot, for obvious reasons, there's not a lot of studies that has been done in human, and few of them actually show increase in TSPO, and one study by Dr. Synchrony here has shown actually decrease in the PBR28 signaling, possibly showing a problem with a dysfunction of the mitochondria. Okay, so indeed what we observe that we observe kind of the activated morphology of the microglia in the hippocampus, but not astrogliosis, because I mentioned the TSPO can be increased also in astrocytes. So I'm not going to show the astrogliosis data, the lack of astrogliosis data, so you'll have to believe me on this. But what I want to show you here is that the male mice were exposed in utero to the anti-caspr2 IgG, show this kind of reactive morphology of the microglia, including less number of processes and increase of puncta of CD68, a lysosomal marker, here in red. And again, this is male, but not female. And if we quantify that using the morphology score and CD68 score adapted from Beth Stevens, you can appreciate the increase in morphology score suggesting these microglia are more compact and they have less processes, so they are more activated, and the CD68 score, more puncta of the CD68 suggesting of activation. And again, if you're using the Scholl analysis to look at the processing length and the function for the distance of the soma, we can actually see very similar, we can see a reduction in those processes lengths. So they are more compact, they are more activated, and this is very basically validating the morphology score here. And again, males, but not female. So let me just briefly stop and do like a 101 on microglia. So microglia are increasingly recognized as important sculpture of the neuronal development. They originate from the myelin progenitor in the yolk sac, and they migrate to the brain around gestational day E9. The microglia development proceeds through 3 distinct stages: the early, the pre, and the adult microglia. And each of those stages, their transcriptome is coincide with the need of the developing brain and its maintenance. So such that the early microglia are more associated with cell cycle, the pre-microglia are more associated with synaptic pruning, and the adult microglia are more associated with immune surveillance. And interestingly enough is that environmental or genetic perturbations of these pathways can alter state-specific function of the microglia, leading to brain alterations. So given our kind of reactive microglia in a model, we hypothesize that suppressing the microglia reactivity following exposure in utero to the anti-caspr2 IgG will ameliorate some or all the ASD-like phenotype and will maybe in the future potentially be beneficial in some cases of autism. So to do so, Ben is an MD PhD in my lab, and he chose to use an FDA-approved drug, an ACE inhibitor named Captopril. And he chose this drug because this drug has been shown to dampen microglia activation in 2 models of neuropsychiatric lupus, and also ACE inhibitors have been shown to improve cognitions in clinical trials in Alzheimer's patients. So how ACE inhibitors might suppress microglia? So there's 2 potential pathways. One is that ACE inhibitor will inhibit the convergence of angiotensin-1 to angiotensin-2. By doing so, there will be less available angiotensin-2 to act on its receptor, and that's going to lead to less pro-inflammation. On the other hand, ACE inhibitors can also inhibit the breakdown of bradykinin to inactive metabolites. So therefore, there will be more bradykinin to act on its receptor, and therefore that will lead to increase in anti-inflammatory. So what Ben did, Ben now used our model. He immunized the mice with Caspar-2, led them to get into pregnancies. When the mice were 3 weeks old, he gave them 2 weeks of treatment, either with Captopril or saline. And what I want to emphasize is that the dosage used for Captopril, which also a drug that's used for hypertension and others, is like 10 times less than what used for hypertension and others. And the reason he uses 3 weeks old mice is because this is, as I mentioned before, a time when synaptic pruning might still happening, and we thought that that might be the critical time window. So now we look on 5 weeks old mice, and we have now a 3D reconstruction, software named Maris. And what you can appreciate here, that the anti-Caspar-2 mice treated with saline kind of show the morphology of the reactive microglia. They are more compact, more puncta, white puncta, and now Captopril kind of ameliorates this phenotype, and it's very similar to what we've seen here with the control mice. And if you look at the morphology score, again, treatment with Captopril suppressed the microglia activation in the anti-Caspar-2 mice to a very similar level of the control mice. And again, in terms of CD68 score, you can appreciate that there is a reduction with treated with Captopril, as you can see here, similar to control. And again, if we use a Scholl analysis, you can see that the compactness, or how the lengths of the processes are reduced in the anti-Caspar-2 saline versus the control saline. But with the treatment with the Captopril, there is more processes suggesting there are more quiescence in microglia compared to the anti-Caspar-2 saline. And importantly, the anti-Caspar-2 Captopril are not much different now with the control saline. So what about the pyramidal cells? Again, this is a tracing now from the CA1 region of the pyramidal cells. You can appreciate that the anti-Caspar-2 saline mice has less branches, has less dendrites, as I've shown before, but treatment with Captopril ameliorates this phenotype, and you can appreciate that it has more complex dendrites now. And this is also now quantified here in the Scholl analysis. You can see the treatment with Captopril now increase the dendritic length. And in terms of the spines, you can appreciate in that image the treatment with Captopril mice treated with Captopril, anti-Caspar-2 mice treated with Captopril have more of spine density, and it's here quantified, showing the treatment with Captopril ameliorate or have increased number of spines compared to saline. So that's probably the most important part, because treatment with Captopril improves the social interaction in the anti-Caspar-2 male mice. So here we use a dyadic play. We put two mice together, we pair mice. They don't know each other, so they are unfamiliar mice, but they have the same immunization and they have the same treatment. And obviously same sex, because now they are all male. And we measure the time the mice are interacting, so they are sniffing each other, they are running after each other, etc. And what you can appreciate here is that treatment with Captopril kind of reverse or ameliorate or increase the time of social interaction compared to male mice treated with saline in the anti-Caspar-2. And each dot here represent a pair of mice, so we give one score for the pairs of mice. So we wanted to show that this is not peripheral effect and mediated through the brain. So what Ben did, he now repeated all those experiments, but this time he used enalapril, because enalapril is a blood-brain barrier impermeable. So, and what he found is that treatment with enalapril, which does not cross the blood-brain barrier, did not ameliorate the structural phenotype in the anti-Caspar-2 male mice. So you can appreciate here that male mice were anti-Caspar-2 male mice were treated with enalapril, very similar to the saline one, and only Captopril ameliorates this phenotype. And again, I've shown you this. This is a morphology score. Enalapril, very similar to saline, but Captopril reduced the morphology score, and the same is true for the CD68 score. And I want to emphasize that these are not historical controls, so this is an additional replication of the data, and the same is true here. You can appreciate that the saline and enalapril treated anti-Caspar-2 mice are very similar to each other, but the Captopril increase the complexity of the pyramidal cells, the dendrites, and this is quantified here very nicely. And again, you can see in terms of the spines that also treatment with Captopril increase the number of spines, but there is no difference between enalapril and saline. Okay, so last we ask, what are the transcriptional changes in the microglia that are induced following exposure in utero to the anti-Caspar-2 IgG, and can we identify the ones that Captopril might reverse or might prevent? So this single-cell RNA-seq, specifically of microglia from the hippocampus, from the same mass that you saw early on the behavior. And I'm not going to talk about this much. This is very much still a work in progress, but I do want to emphasize here on the UMAP that you can see that many of the cells here, these are more, what you can see is that many of the cells are here are more quiescent microglia, and when we move in the UMAP, these are more the reactive microglia. And if we now do the CNA analysis, basically a fancy way of looking at distribution of the cells, you can appreciate that when we do the analysis between Caspar-2 saline versus the control saline, there's less cells here in blue from the Caspar-2 salines and an area where the more quiescent microglia, but there were increase of the number of cells in the area of the more activated microglia. And if we now look at the differential expressed genes between the Caspar-2 saline and the control saline that are actually reversed by the Captopril treatment, okay? So if we now look at the number of differential expressed genes that were upregulated in the anti-Caspar-2 and were downregulated by the Captopril treatment, we see that many of those cells many of those genes are actually not ameliorated by the Captopril treatment. However, if we're looking now at the number of differentially expressed genes that were downregulated in the anti-Caspar-2, but they were upregulated by the Captopril treatment, you can appreciate there's many of those. And interestingly enough, about, I would say, almost 90% of them are actually here in cluster number 2. Most of the genes that were downregulated in cluster number 12, as you can appreciate it here, they are now upregulated by the Captopril treatment. So I'm not going to talk about much of what is cluster 12. We'll save it for the next time. But I do want to draw your attention for a possible conclusion from this is that from this side, when we see many of genes that were upregulated in Caspar-2 but not downregulated by Captopril, we can think that maybe restoring the social behavior can be achieved even when not all genes are actually reversed. However, that downregulated genes, because we see many of those downregulated genes were now upregulated back by Captopril, maybe those are the genes that we need to focus in because they might play a significant role in the autism-like phenotype. So I'll end up here, and I hope I have convinced you of the importance of the in utero environment, and specifically the contribution of maternal antibodies and anti-Caspar-2 IgG to brain development and to autism. We think that continuous microglia suppression may suppress autism-like phenotype at the right time window, and that reactive microglia can be therapeutic target. ACE inhibitors are only one therapeutic modality, and there are others that we can discuss. And most importantly is that the autism phenotype can be modulated even after the insult to the brain has occurred. So I have a really big team of cheerleaders here, and I presented most of the work of Ben here today, who is an MD PhD in my lab, and Sierra is my postdocs. Sierra, I started all this work back in the day when I was postdoc, and all the other fantastic mentors and collaborators. So thank you for inviting me, and hope I have time for questions.
Thank you. Yes.